Okay, let's continue. Now, when we discussed about stem cells, we said they are, this is just a general, sorry, graphical representation of stem cells. So there are cells which are uncommitted cells, undecided cells. Then there are cells which are committed and determined cells. And then there are cells which are differentiated, and that's the final function. Stem cells lie somewhere between uh, committed determined cells and undetermined cells. And here you can see the potency, it increases with age. The more the cells specialize, the less potent they become, and it also increases with time. That's age. Skin cells, they actually divide very slowly, and as I said, they have variable potencies. They are somewhat committed. When we say somewhat committed, what is meant by somewhat committed? Because we previously discussed, we talked about committed versus uncommitted cells, and now we're talking about somewhat committed. So what are somewhat committed cells? We will discuss in this representation here. So we have a stem cell here. What happens that this stem cell is very slow, and it divides. It divides into another stem cell. It also needs a proper appropriate stimulus. So under the correct and appropriate stimulus, chemical regulators, this stem cell will divide, and it will give rise to another stem cells and a progenitor cell. Now, progenitor cell, as the word says, progenitor, it gives rise to progenies. And when this happens, the progenitor cells, this stem cell, it will still be very cool and calm and slow dividing, but this progenitor cell, it will quickly, so this arrow represents only the progenitor cell, it will quickly and uh, divide a lot. So a number of progenitor cells will be produced. Now, when we have these different number of, or a large number of progenitor cells which are dividing rapidly, when they are produced, what will happen? The progeny of these progenitor cells will go and differentiate into one or more than one of different types of cells. And you could see, so like now they have been developed into more differentiated cells. So here, this cell is pluripotent at this stage because it has many fates open to it. Now, when the progenitor cells, these progenitor cells, they are multipotent. They can give rise to a number of organisms. But with these cells, they are unipotent or bipotent because they are more differentiated. So when we say more differentiated, then this is then they are when we say they are more differentiated, then they have less potencies. One thing that I could say about progenitor cells, when we say stem cells, they divide very slowly and they have variable potentials and they are somewhat committed. So what about progenitor cells? Progenitor cells are also somewhat committed, but they are more committed than the stem cells because you know that they know which lineage or which uh, specialized cells they were going to develop into. And the number of different cell types that originally uh, that originally these number of different cells that originally coming from a single progenitor cell is a reflection of the different potencies of stem cells, and it in itself is a very interesting and attractive phenomena that one stem cell is actually giving rise to all these different types of cells. And this kind of a diagram of stem cell is called lineage. So this is the lineage. This whole figure shows the lineage of stem cells, and the diagram is known as the lineage diagram of stem cells, where uh, a set of cell types arise from a single or from one single stem cell or one progenitor cell. So you could also take in consideration for the lineage of stem cells, this stem cell, this progenitor, because even if this stem cell divides, it will give rise to another progenitor cell. Okay, this is actually the same figure discussed in case of hematopoietic stem cells. You know, there's a hematopoietic blood stem cell. Now, this is actually the pluripotent cell. This will divide into, now this stem cell will divide into a progenitor cell and a stem cell, but that is not shown here. What we see here as only progenitor cells. Now the progenitor cells, they are multi-potent. This is a pluripotent cell and they are multi-potent cell. And you can see it could go the myeloid stem cells to make red blood cells, to platelets, to myeloblast. And this myeloblast can then go and make eosinophil and neutrophil and basophils. Similarly, the lymphoid stem cell, this is again a progenitor cell, which is a pluripotent cell. This is a multipotent cell. This could make lymphoblast. It could go for B lymphocytes, T lymphocytes, and the natural killer cells. So when we talk about lineage, stem cell lineage, then the hematopoietic system gives us the best, one of the best pictures that is easy to understand about the stem cells and their lineage through progenitor cells, where we could see the reflection of different potencies of the stem cell. Discovery of stem cells, it's not easy actually. The important question that was always arised, if or if not a body can heal itself. We know it for a long time, that in the early embryonic stages of the development of a zygote, of an embryo, there are repeated and a number of cell division events which give rise to other new cell types or organs and eventually whole organism. That is all understood. The thing that was not understood, what happens when the adult phase is reached? So what happens? How does the turnover of the tissue and the whole process of maintenance adult fate works? Is there a maintenance system? Is there a system through which the adult body can heal itself? And different techniques were used. One technique that really gave us all the answers was known as turnover assay or pulse chase assay. It was used to determine the cell turnover to get the half-life of the cell. And this would tell us how old these cells are and how long would or could they live. Okay, now this is actually a figure that tells you the pulse chase experiment. In this pulse chase experiment, what we do, we have a cell population growing, and then you add BRDU. Now, what is BRDU? BRDU is actually a bromodeoxyuridine. So this is this is actually an analog to deoxythymidine. Thymidine is the G in, it intercalates into the cell, and then you give a very short pulse, like incorporate into a DNA, but it gives a very short pulse of it. You have the cell labeled. Now you can see after a short pulse, cell population is labeled, and then add lots of unlabeled thymidine follow cells. So now you have labeled cells and you have unlabeled cells. And what you do, you chase them. You chase means you follow the cells over time and you find out which cell is surviving and how long it is surviving and which cells are dying. And this way you can assay the half-life of these cells, and the half-life and the turnover assay. The turnover means like dying, cell dying, death of cells. It gives us a very important, they gave us very important findings. So the turnover or the pulse chase experiment revealed the cells, they do not live forever. They do turn over, which means they die. But this is a very interesting point, that they are replaced by new cells. They do die, they do not live forever, they turn over, but there is a replacement system. And the to put it in a nutshell, the important points we got from this essay was there is indeed a replacement system in our bodies which repairs and maintains our body organs. And we found out that this very system is called the system of stem cells, or stem cells are the system which replaces and repairs and maintains our body. Now, from the Pulse's experiment, we got another proof that all organs, they will show turnover. I mean, they will be exposed to turnover, they die. They will go to die. For example, an RBC, a red blood cell, it takes about 120 days to die. This means if an RBC is, one RBC is dying after every 120 days, 10 to the power 7 new cells have to be made every day, and there has to be a biofactory for this. How do you replace? This is a replacement. So 10 to the 7 new cells are replaced by the hematopoietic stem cells. This is how we say that the system for repair and maintenance is stem cells. Similarly, small intestine. And now if you see the half-life of small intestine, 4 to 5 days. And in small intestine are very important because all the food, everything that we eat, important salts and nutrients and other stuff, it will absorb in the small intestine, in the structures, the finger-like projections, they are known as villi or villi, singular is villus, figures coming out in a minute. And then here on a human head, 4 years. So if there are no stem cells for the production of hair, you would be balding after 4 years. But there are actually, there is a replacement system. So stem cells are everywhere in our body. They are not something out of this world. Now this is a figure of small intestine. This figure, the duodenum, the jejunum, the ileum, we are not interested in this, the stomach, and then the large intestine, and eventually the small intestine. This is where we are interested. That's the lumen, there's a mucosa, submucosa, we don't care about this. The most important piece of information that we need is here. And you can see this is where all the digestion, the absorption of the food and the nutrients happen. Right? These finger-like projection villi, and they provide a number of, since they are microscopic projections. So the smaller the size, the more the surface area. That's why nature has designed in this way so that more and more surface area is provided and more food is absorbed. And this is another exemplification figure of the villi in a cut cross-section area. The important figure is this one. So these cells that you could see, these ones, they're lined, these villi, these finger-like projections, these are the cells which die after 4 to 5 days. So each of these cells has a half-life of 4 to 5 days. And if they have a half-life of 4 to 5 days, which means there has to be a replacement system. If we do not have a replacement system after 4 days, all of these cells will die. And when they die, we will not be able to digest any food. Turns out there's a structure in small intestine in these finger-like projections, which is in this cavity known as crypt. And this crypt is the place where the stem cells for these small intestine are residing. It's not easy to find them. It's very difficult to find them. Why? Because they divide very slowly. Why? Because they are very rare. They are very rare and they are very low in number. But about small intestine, we know that these guys are somewhere in this crypt area. So this is another junction of the villus, another figure of the villus where we have this crypt. And these different color, not yellow, kind of gray colored cells are speculated to be the stem cells. So what happens when the cells die? these guys will produce new cells and they will push the cells upwards. If a cell die here, there will be a new cell produced here and they will push these cells upward. So the dead cell would be replaced by a new cell. How and why are we sure about this process? Because in a pulse chase experiment, this was done. So they were using some kind of stains, and you could see there's the crypt. So these are the cells, and now it's a time. Let's suppose the time 0, it's time 1, time 2, and time 3, and you could see the movement of these cells. These cells are time 0 here, but at time 1, after some time, they are pushed up. So which means that there is a replacement, some replacement of cells coming, because these cells, they're moving. You see, they're moving among this line. They are here, and in time 1, they're even more further, and in time 2, so 0, 1, 2, and 3. And in time 3, you could see they move all the way from here, which means there is a replacement system, which means there are new cells are added here, and when new cells are added here, they just pushed upwards so that the old cells could be replaced.